Good afternoon, everybody. Welcome to the presentation of the 21st Annual Gruber Neuroscience Prize, on which the Society for Neuroscience is very pleased to collaborate. The Gruber Foundation presents 3 annual $500,000 prizes in the fields of cosmology, genetics, and neuroscience. Sarah Rea, the Executive Director of the Foundation, will say more. Sarah?
Thank you, Professor Picciotto. We are delighted to present the Neuroscience Prize at SFN's 53rd Annual Meeting and to continue to work with the Society. The Gruber International Prize Program, established in 2000, recognizes achievements and discoveries that produce fundamental shifts in human knowledge and culture. While we are here to honor Cornelia Bargmann and Gerald Rubin, let me mention that they join Cosmology Prize recipient Marcia Ricci and Genetics Prize recipient Hugo Bellin on our 2024 roster. Please note that nominations to the 2025 Gruber Prizes are open until December 15, 2024, and that we encourage nominations that reflect the breadth of the fields and the diversity of those working within them. Before we return to neuroscience, I must acknowledge our co-founders, Peter and Patricia Gruber, whose combined vision and leadership established the International Prize Program, and whose care in doing so gave it the legs to stand on its own. We are grateful to SFN for its support of the Gruber Prize, but also for administering the International Research Award in Neuroscience that we fund. Let me now ask Marina Picciotto to present this year's fellows.
Thank you, Sarah. It's now my honor to present the Peter and Patricia Gruber International Research Award in Neuroscience. This award recognizes up to 3 promising young neuroscientists for outstanding research and educational pursuit in an international setting. This year's recipients are Dr. Angelo Forli, Aditya Nair, and Kami Testar. Dr. Forli is a postdoctoral fellow at the University of California, Berkeley. He received his PhD from the Italian Institute of Technology under the director of Dr. Tommaso Fellin. In Dr. Forli's investigation into how animals navigate in collective settings, he developed an innovative system that combines precise tracking of large groups of free-flying bats, it's cool, right, with wireless neural recording methods. Beyond spatial coding, he found that the hippocampus tracks and maps the presence of other bats, their location, identity, and sensory cues of other conspecifics, enabling successful navigation within the group. The award selection committee was impressed by Forli's creativity in combining complex natural social behavior with neurophysiology, his remarkable publication and funding records, and his stellar letters of recommendation from leaders in the field. Please join me in welcoming Dr. Forli. Our next recipient is Mr. Aditya Nair. Mr. Nair is a PhD candidate in computation and neural systems at Caltech, and he previously studied in both Singapore and in India. Nair's interdisciplinary work combines neurobiology and machine learning to explore how the brain controls social behaviors that are disrupted in a wide range of developmental and mental health conditions. His computational expertise enabled him to provide the first demonstration that an aggressive state in a mouse is encoded in the emergent dynamics of neural populations in a brain area called the ventromedial hypothalamus. This finding represents a new way of thinking about the hypothalamus and suggests that a common network computation is used to encode diverse affective states. The award selection committee was impressed by Nair's breadth of international experience, as well as outstanding publication record at such an early stage of his career. Please join me in welcoming Mr. Nair. Our third recipient is Dr. Camille Testard. Dr. Testard is a junior fellow at Harvard University. She completed her PhD last year at the University of Pennsylvania under Dr. Michael Platt. Dr. Testard's work has focused on the social behavior of macaques as seen in both in the field and in the laboratory. In her field research, she examined the social behavior of macaques on a small island in Puerto Rico after Hurricane Maria. In the lab, she recorded neural activity of male-female macaque pairs using minimally invasive devices, allowing for more naturalistic behavior. The award selection committee was impressed with the uniqueness of the work and with Dr. Testard's commitment to incorporating natural social behaviors in a science dominated by laboratory research. Please join me in welcoming Dr. Testard.
Congratulations to the 2024 Fellows. We hope to see you back here soon, perhaps receiving your own Gruber Prize. The 2024 Neuroscience Prize recipients were chosen by our Distinguished Selection Advisory Board. These volunteer advisors bring their priceless expertise and commitment to the selection process, along with an irrepressible enthusiasm for science. They are nominated to serve by the Society for Neuroscience. We could not do this without them. Let me now ask our board chair, Frances Jensen, to read the official citation to the prize drafted by this Selection Advisory Board and add any further remarks.
Thank you very much, Sarah. Well, it is an enormous privilege to read the prize citation as follows. The Gruber Foundation proudly presents the 2024 Gruber Neuroscience Prize to Cornelia I. Bargmann and Gerald M. Rubin for their fundamental research and leadership in the use of invertebrate genetic model organisms, C. elegans, Bargmann, and Drosophila melanogaster, Rubin, in neuroscience. Both investigators have done rigorous and important fundamental studies relevant to gene function and the organization of circuits in behavior. Both investigators have developed new genetic tools, methods, lines, reagents, which are used today by the entire field. Thus, Bargmann and Rubin have facilitated, accelerated, and democratized scientific research and have created vibrant international communities and educational opportunities using these genetic model invertebrates. So it is my pleasure, before they come up, to just comment on and reflect some of the discussions of the committee as we arrived at the conclusion that these are our two prize winners. The work performed by Bargmann and Rubin has advanced the field of neuroscience in multiple ways. This includes the work they've done that has offered insight into the question of the link between biology and behavior, as well as their generosity in creating and sharing tools that have helped move the field forward by offering new capabilities for carrying out research accessible to a wide range of institutions. Their impact on the field cannot be understated. Dr. Bargmann is being recognized for her role in linking genetics with innate behaviors, but also the underlying dynamics of small circuits, neuronal populations, and molecules. Her work has also determined how the environmental cues interact with genetic variation to produce flexible behaviors. In addition, Dr. Bargmann is a consummate mentor, advocating and serving as a leader in some of the most impactful organizations in our field, such as the Chan Zuckerberg Initiative and the BRAIN Initiative. She has championed support of basic science tools, tool development, and application of this research. Dr. Rubin is being recognized for his role in developing a host of tools and resources that have advanced the field of Drosophila neuroscience and undertaking the massive venture of mapping the fly brain. This work is has identified the constituent neurons and their connections to each other, all of which have enriched the field in a number of invaluable ways. Like Dr. Bargmann, Dr. Rubin is a leader, an exemplary mentor for the field. Over his tenure and leadership at Genelia Farms, he has prioritized both developing new tools and models, but also disseminating them widely. There are so many reasons that the prize committee felt this should be a shared prize. Both scientists developed, led, and advocated for the principal concepts regarding the value of fundamental neuroscience. The conceptual frameworks they each developed have gone on to support and seed neuroscientific hypotheses addressed in other organisms and in humans. They have shared freely and generously throughout their careers. And finally, the committee recognized how their tools have allowed world-class science to be performed in a wide range of institutions, increasing the accessibility of neuroscience for all. So, Cornelia Bargmann and Gerald Rubin, please come forward to receive the Gruber Neuroscience Prize.
There will be a moderated discussion with the recipients after their lectures, and that discussion will be moderated by prize advisors Eve Martyr and John Monsell. Thank you.
I'd like to thank the Gruber Committee for honoring me and my lab's work. I appreciate that they paired me with Gerald Rubin, a scientist that I respect and admire, and with whom I argue on a regular basis. It's perfect. I would like to thank the Gruber Foundation for supporting this prize and providing an opportunity for science to step back and celebrate advances here in the neurosciences, and for us to share them with each other and for the broader public. Science can be slow and hard, and it's wonderful to have a chance to celebrate the wins. I had the pleasure of meeting Patricia Gruber, and I just want you to know that she is a psychotherapist. She is one of us. I also appreciate the Gruber Foundation and Patricia and Peter Gruber's work for their prizes in other areas, including genetics and cosmology, and for their support of human rights and women's rights, and the value that that has in the world. And finally, I want to point out that the committee really represents a very broad and thoughtful group of neuroscientists. And in the past, they have acknowledged neuroscientists in areas ranging from molecular neurobiology, neurophysiology, development, systems neuroscience, theory, and disease-relevant research. And through that example, they represent, along with the Society for Neuroscience, that inclusive principle that neuroscience is one endeavor, and that we are all engaged in a common discipline, and that nothing could be more important or inspiring than understanding the brain. Thank you.
I'd like to join Corey in thanking the Gruber Foundation, as she did, and of course, I'd like to thank all my many mentors and the talented people that worked in my lab, my wife for 47 years, and the Howard Hughes Medical Institute, who supported me generously for 38 years. I guess we don't need these slides. Sorry, I'm just. So what I would like to talk about today is connectomics, illustrating what its importance is and how we're going to increase its power in the future. So when I got involved in this field, coming from being a molecular geneticist purely, 15 years ago, I put together this slide which showed the way I viewed what we had to do to understand the brain. A pretty atypical view for most neuroscientists. We needed to determine anatomy. We needed to develop methods to genetically address the individual nodes in a circuit so we could see what their functions were. We needed to determine those functions by inactivating cells or changing receptors within cells, and then doing physiology on those circuits. So in the first third of my talk, I'm going to tell you about what we've done to try to build these tools, in particular these first two tools, which are the ones that I've actually worked on. So anatomy. So coming from just having finished the genome project, I was very much into having maps. I thought we needed to have a detailed wiring diagram of the fly if we were going to have any hope of understanding the brain. This was not, especially in 2010, a very popular idea in the neuroscience community. Most people thought it wasn't feasible, or even if you could do it, you wouldn't learn anything. And we certainly didn't know how to do it. We knew that we'd have to improve the techniques by more than 1,000-fold, even to do the fly brain. We had no real idea how we were going to do this, but we were convinced that without knowing this, we would never understand the fly brain at the level that would be satisfying to a highly reductionist molecular geneticist like myself. So because this was an interesting challenge, we had no trouble identifying a team of talented people who were willing to bet their careers that we could make this work. And maybe most importantly, we're funded by the Howard Hughes Medical Institute, who wanted us to do high-risk, high-reward problems and was willing to put up the money, to date $60 million, to complete this project. So our first accomplishment was a teeny piece of the visual system, 2013, then a slightly bigger piece, then a piece of the Learning and Memory Center, the mushroom body in 2017, and then 2020, actually 5 years ago, I gave a talk from this podium about completing the connectome of this part of the brain, which we call the hemibrain. Now, even though this isn't the whole brain, we chose carefully to include the two most cognitive parts of the fly brain, the mushroom body, center of learning and memory, and the central complex, center of spatial navigation. And when this connectome emerged, it was analyzed in great detail by neuroscientists, and we attracted theorists who were able to model this and make models that were constrained by having a wiring diagram. So that was a new development, and we learned a lot of important things like how flies do goal-directed spatial navigation came out of those studies. So for me, this was the proof of principle, and I thought it should be enough to silence the skeptics, which it did silence some fraction of them, but probably not all of them. Now, just last week, there was a whole series of papers on a larger volume that covered the entire female brain above the neck that was published in a series of papers. And after the hemibrain was done at Genelia, we turned our attention to the male brain. All these other volumes were female brains. And we released recently the visual system of the male brain, and in a few months, we'll release a connectome of the entire male brain. And this will include not just the stuff above the neck, but the stuff below the neck, the fly spinal cord, which you can see is quite sizable, and the connections between the brain and the spinal cord. So this will allow us to compare male and female brains, which we expect to be a few percent different, and also trace information flow from sensory systems to motor neurons. Okay, so there are many other projects on connectomics that I'm not have time to go through, but I think we're in pretty good shape for anatomy at the level of the connectome. But then the tools to develop things to manipulate the nodes in the circuit That was my real concentration because I was a molecular geneticist. And 2 years after Genelia opened, we published this proof of principle paper that we could take individual fragments of the genome and use them to drive highly specific patterns in the fly brain, patterns that were well under 1% of the cells. And we actually made 8,000 of these lines, and they've been very heavily used, over 1,000 publications. But as far as I was concerned, we had failed. And the reason we had failed was these were not cell type specific. These were still populations of cells. Small populations, but populations. And the way to solve that problem came from doing intersections between two enhancers. The idea is, if we take these patterns and we look for two enhancers that only overlap in the specific cell type, we may be able to get that next level of specificity. And that's what happened. So this shows an example from 2014, where we have two patterns driven by two different enhancers. And then when we intersect them, we get this pattern, which is a single cell in each hemisphere. So this is 1 in 100,000 specificity, single cell type. Okay. So we went on and we made these collections of cell type specific lines for lots and lots of parts of the brain. This just shows a set from the visual system. And at this point, we have them for over 1,000 different cell types. So I'd like to declare partial victory here and say we can do this certainly for most cell types of interest if you put in the time and effort. And then many other groups developed behavioral assays and methods for optogenetic manipulation and calcium sensing and other things. Okay. So now I'm going to move into the middle section of my talk, which is applying these methods. And the example I'll use is female-female aggression. Most work in flies has been done on males, and our first forays into aggression were in fact done on males because a number of years ago, in 2013, Eric Kupfer, who was a postdoc with David Anderson, came to Genelia to use our genetic resources to try to find lines that, when activated, would induce male-male aggression. And he found such lines, and it's shown here. They're male-specific neurons, and when they're activated, they can induce a persistent aggressive state in males. And that is shown in this experiment, where flies were separately optogenetically activated. After a 10-minute cooling off period, the divider between them was removed, and you see they're still quite aggressive against one another. So this was our first foray into aggression, and then Eric went back to Caltech to finish up the studies, and I thought I was totally done with aggression. But then, 5 years later, by accident, as part of another study, we discovered a population of neurons in females, female-specific population, which would induce aggression in females when activated and a persistent aggressive state. So I'm going to refer to these as aggressive state neurons, and they're always going to be colored this color orange. And about this time, a new postdoc arrived in the lab, Katie Schroeder, and I said to Katie, this is a great project for you. You're interested in behavior. You figure out how this works. So Katie's very interested in social behaviors and would like to understand how they work. And so she set off on this project. But I want to say this was not at all my motivation. My motivation as a tool builder was to know how good our tools were. So to do that, you have to test them. So the test was, we have a neuron population, 1,000 cells, in the middle of the brain, 5 synapses away from any sensory input. And when they're activated, they induce this aggressive state. So could we explain how these neurons are making the female flies aggressive? That seemed like a good test. So any complex trait would have done for me. This was just convenient. And females, this shows females fighting at slow motion, and females have their own style of fighting, like this head-butting maneuver that males don't really do. Here's a group of females, the way they behave with one another. Before this neuron's activated, they'll touch one another, but they pretty much walk away. When you activate this neuron, you'll see the flies will cluster, and they'll start fighting with one another. And you can see this in a higher magnification here of them doing this. So our first clue into how this happened was by the observation that when blind flies are placed in a reader and this neuron's activated, they just touch one another, bump into one another, but they walk away. They don't fight when they're blind. So this led us to think about this as maybe this neuron was affecting how the brain focuses its visual attention on different features. And this is, of course, a problem that any visual animal has. So when you're driving down a crowded highway, you need to know to focus on the car in front of you and not be looking at the scenery if you want to do that maneuver safely. So flies, when they're fighting, need to pay attention to other flies that are moving in their natural environment. And we found that this was exactly what this neuron did, and it did it by 3 different mechanisms. And for time, I'm really only going to be able to tell you about one of these mechanisms, the top one, which we were calling a toggle switch. So what this circuit does is shut down information flow through the population of visual projection neurons called LC10C and simultaneously turn up the flow through a different population of visual projection neurons called LC10A. And LC10A is specifically tuned to detect other small moving objects, that fly-sized small moving objects in the visual field. And so we did all these methods I told you about. This is just an example of a behavioral test. So in this case, we inactivated LC10A, and aggression went way down, and we inactivated LC10C, and there was no effect on aggression. So this is consistent with the model. So here's this switch again, and now I had to put some neuron names here. And the LC neurons, the visual projection neurons, there are about 100 of them of each type because they have to tile the visual field. And there's about a dozen of these aggression-promoting neurons, but the other neurons in this circuit are 1 copy per hemisphere. So if we look at the lower arm of this circuit first, what we see is the aggression-promoting neuron, the orange neuron, activates this SMP54 neuron, which is GABAergic, which then inhibits the pink neuron, which is normally inhibiting LC10A. So when you activate aggression neuron, you disinhibit LC10A. And by doing patch clamp electrophysiology, May, our lab electrophysiologist, was able to show that when you activate the aggression neuron, you depolarize, you hyperpolarize the pink neuron. That was just expected. And the other thing you can get from connectomics is where all the synapses are. So I just want to point out that the neuron that's repressing flow through LC10A sends its synapses to the axonal termini, intermingled with the output synapses of the visual projection neuron. So it's shutting down its ability to communicate with any downstream target. And the other arm of the circuit, the top arm, is much simpler. It's just the aggression neuron activating this purple neuron, which you can see here in the electrophysiology. Okay, so that, at this point, I've shown you a little electrophysiology, a little behavior, circuit mapping, anatomy, and now we wanted to ask the question of, does this circuit also function in males? Because the only sexually dimorphic neuron in that circuit I showed you was the aggression-promoting neuron, the orange neuron. And the other neurons were there in both males and females. And so we thought maybe this is a general switch that allows flies to focus on other nearby flies, say, used by males during courtship. And there was a nice study for looking at this based on work done in Vanessa Ruder's lab. And they had shown that during courtship, males will chase a small fictive dot that they think is a female moving in their environment. And they'll get very excited and they'll fixate on that dot and trace it. And then when they do that, simultaneously, information flow is turned up through LC10A. So it seemed obvious to postulate that the same toggle switch was operating in males. This driven now has to be some other neuron because the female aggression neuron isn't there in the males. And so we set out to test this. And we tested it by imaging, Tom, Vanessa's student actually tested it, by imaging from this purple neuron, which we'd expect to go up when the males are courting a female, and the pink neuron, and we expect it to go down. And this is exactly what Tom saw. The black trace is just when the flies are running around the ball. There's nothing about motion here. But when the target site's oscillating, you see that the activity of the purple neuron goes up and the pink neuron goes down, as you would predict from this model. Okay, so this is all great, and these are the people that did this work. I mentioned them during the talk. But before I conclude this part of my talk, I want to say that there's something else we want to look at, which was how do flies disengage? So flies do not fight to the death. As soon as it's clear that somebody's going to lose, the loser retreats to live to fight again another day. So what shuts this down? Well, this is a case which I loved because we could see what was going on just by looking at the connectome without doing any experiments. Because what we found was there was a neuron that you might think you might do this simply by that neuron just repressing this aggression-promoting neuron. But instead, we found there was a neuron that went to all the targets, all the major targets of the aggressive neuron, and repressed them, providing an input of opposite sign to them, as shown here in this diagram. And so, for example, here in this neuron here, the purple neuron is getting input from the aggression-promoting neuron and also from this disengagement neuron. And those are just the number of synapses, those numbers. And then this other neuron I talked about, this GABAergic neuron, also gets input from both of these. And that's true for, you go down the list of the top 20 targets of the aggression neuron. Okay, so this seemed to us to say what might be going on. And of course, since we had the connectome, we knew what this neuron looked like. So there's a single neuron on either side of the brain. So here is where I want to illustrate what you get from the connectome and what the connectome allows you to do in an organism like flies with our other tools. So from the connectome, we can tell who's downstream of this neuron, and we can also look who is upstream of this neuron to get an idea of what clues in the environment would tell a fly it's time to disengage. But also, since we had the neuron, we knew its morphology, and we had image data from thousands of enhancers. So we could search that database to find enhancers that would drive expression in this neuron, find two of them, intersect them in the way I mentioned before to make cell type specific neurons, which we did. And then Katie used those to show that when you activate this neuron, you could stop flies from fighting in females and also in males. So now we have the neuron, and we're all set up to do the circuit. And this is something that I hope Katie will do in her own lab, because I want to stop doing this kind of research, because I'm convinced that my job as a tool builder, I've always been a tool builder first, is done. Because we've used all these tools successfully, and I've shown you that they're synergistic. The connectome is only a tool, and its power is only revealed when used in connection with other tools or when analyzed by theorists. All right, so I'm going to conclude by talking about what we're missing and what I think needs to be done next. Okay, what tools do tool builders like me, aging tool builders like me, need to do in our remaining time as scientists? So I actually learned a little bit of neuroscience during this 15 years, so I knew what we were missing, or some of what we were missing. So, for example, we can see chemical synapses in the EM pretty well, but we can't tell what neurotransmitters they make. And there's been some progress in machine vision to do this, but I would say that's incomplete because there are a lot of rare neurotransmitters that aren't predicted, and there's a lot of cases of co-transmission. So we could look at that. And then we wanted to study neuropeptide-mediated signaling, something that Corey has worked on for a long time, and Eve Marta keeps reminding me that we're not done until we look at the neuropeptides. So we decided to have a look at the neuropeptides. And as this audience all knows, neuropeptides are made as precursors, which are processed and cleaved up in dense core vesicles, which are distributed across the cell. So the first thing we had to do was survey all the neuropeptides in the fly. From the genome sequence, there are about 40 or 50 predicted that will be in the adult brain. And we first looked at them by RNA in situ hybridization to ask what cells express them. So this is the expression of the PDF neuropeptide, one of the most heavily studied neuropeptides in the fly. And it's just expressed in a small number of cells. And here's the other extreme of peptide called small neuropeptide F, which is expressed in about 100,000 cells of the 100,000 cells in each brain hemisphere. So this is the other end of the extreme. Okay, so this just gives you the number. We want to know what cells express them. So one approach to that was to label the in situ hybridization, but to fly brains where a cell type was marked. And we did that with a large collection of lines that we had made for the central complex. And this is an example of one such cell type there. And it looks like this in an EM section. So I mentioned earlier that after the hemibrain, there were some very detailed studies done. So I want to point out that this figure is redrawn from panel B of figure 56, figure supplement 1 of this paper. So these were, in fact, detailed papers. But the important thing is we didn't detect any expression for those neurotransmitters or for 15 of the 17 neuropeptides we tested, but we found expression for 2 neuropeptides and 1 rarely used neurotransmitter, tyramine. And I'm just going to show you a little bit of experimental data. This is one of these in situ hybridizations, and this cell type is marked in green. You can see the outline of the cell bodies and the processes. And you can see here that this cell of this type is expressing the enzyme that takes tyrosine to make tyramine, but it's not expressing this other enzyme you need to make tyramine into octopamine. So these cells make tyramine as a neurotransmitter, and then we could take all our neuropeptide probes and hybridize them against this genetic background, and we can see that these cells made 2 neuropeptides, which are shown here. So once you can do this, you can do a lot of it, and this is 120 cell types in the central complex, about 40% of the central complex, with what cells they express. And about half of them express neuropeptides. It was known to be a peptidic region. All the ones that express neuropeptides pretty much express a small neurotransmitter. There's lots of examples, co-transmission. So it's sort of interesting from that point of view. But we're just looking at transcription, and when you really want to look at the action, it was in the dense core vesicles, the neuropeptides. So to do that, Thomas Ravencroft, a postdoc in the lab, started to epitope tag the neurotransmitter precursors in the part of the precursor that's not going to become the stable peptide. And when you do that for PDF, you can get an image like this. So in the magenta is the RNA expression, and in the green is the dense core vesicles that are made in the cytoplasm and distributed throughout the axons of the cell. So we wanted to do that for all the neuropeptides, and we're about halfway done for doing this for all the neuropeptides. So this just shows SNPF and part of the brain. This is so dense, if I showed you a total projection, it would be meaningless. And of course, we want to know not just the neurotransmitters, we want to know the receptors. So this is a picture from the same part of the brain of a tagged small neuropeptide F receptor. So you can just see by looking at these, it looks like it could be local volume transmission, but we have to do this for all the other receptors. So we're happily, as tool builders, doing this for the 300 or 400 proteins of interest in the nervous system so that we can provide these as tools for others and try to map them. And then we're looking at gap junctions or electrical synapses. These are what these look like. They're called, the subunits are called the annexins in flies. They're like the connexins in vertebrates, and there are 8 of them in the fly genome. And they all look very similar in structure. And I'm just going to show you data for the one that's the major one in nerve cells, which is shown in this low-resolution movie of the fly nervous system. You can see it's all over the place, but it's not everywhere, not in every cell. And you can see this at higher mag here in a portion of the brain where you're seeing where the synapses are in magenta and where the gap junctions are, or where these channels are. Some of them could be hemichannels, some could be gap junctions. But you see there are some places where they're collected together, and they're probably, these rafts are probably real gap junctions. So of course, we want to map where the gap junctions are. But here's a problem. We can mark the cell like we did for the neuropeptides, and we can say this cell expresses gap junctions. But we not know who it connects. It's not just a single cell doing it, it's a connection between two cells. So we have to mark the other cell. And we have tricks for doing this with different drivers. So we can do something like this, and I'll show you an example. This is a giant fiber, the escape neuron in flies, which is known to use electrical transmission. And in green and in the magenta is one of its downstream targets. And in the blue is the Shaker B protein. And you can see they're lined up, and it looks just like you would imagine it should look like. The problem is this doesn't scale. If you think about having to make driver lines for every cell type in different colors, it's just non-doable if you want to do it for everything. And we want to do it for all cells in the brain. That's the goal. So what you'd really like to do is take advantage of what you have in EM, where you label all the cells. They're all there, and then you have to, and then you can map them. So if you could do this, you could map all the gap junctions, you could map neuropeptides, you could tell when a neuropeptide was at the membrane being released. So we really wanted to do that, and fortunately, this method was just developed for doing this, and the method's called LICON, and this is the reference for the papers on bioRxiv now. I expect it will be published soon. And Mo, who was the first author of this paper, helped my colleagues at Genelia set this up. This is data done by Josh Lilvis at Genelia. So this is what a piece of a fly brain looks like, staying this way, and Josh could trace the neurons in this body, and this is a piece of the brain, and we could stain this with antibody for synapses, and this is what the synapses look like. He could predict from the from just the gray image where the synapses were using machine vision, and show by overlap that those agreed, and then you could see them, the synapses marked here, and you can see opposite them densities that look like postsynaptic densities. So this is all great, and at Genelia we have all sorts of strange and wonderful microscopes, and we use these two microscopes, and okay, now here we have a image, and I just want to point out the resolution. We're using a microscope, a super resolution microscope, and we're expanding 20 times. So the voxels in that image I just showed you are 5 by 5 by 10 nanometers. That compares with EM resolution voxels. So we're getting EM resolution in the light microscope. So now with all these antibodies and TAC probes, we can map all these proteins. This is a mirror microscope, which has bigger voxels. So it's not quite there, but it's a very exciting instrument. So this is it imaging the entire fly brain. The fly brain now is 8,000 times bigger in volume. It's literally the size of a mouse brain. And this mirror microscope produces images like this. They're not quite the resolution we need, but they're still pretty attractive. But the important thing here is it images a complete fly brain in less than an hour. So those of us who care about technology and knowing we need orders of magnitude more to get to bigger brains, this becomes important. I'll show you this is what the mirror microscope looks like. There was only one in the world. It wasn't built for this purpose. It was built by Philip Keller. It's called the mirror microscope because mirror in it, and it has very big lenses, you can see compared here. Okay, so there's where I'm going. I hope over the next few years I can work with my colleagues to add all the protein information to the brain connectome map, provide the tools. Other people will want to use these. Of course, when we have learning and memory and other changes, aging, whatever, things are going to change. A lot of that's going to change at the level of the proteins that you'll need tools like this to visualize, not just what you can see in EM connectome. And then my lab is going to go off and try to solve some of the many mysteries of how gap junctions and neuropeptides are trafficked and arranged and what their structures and associated proteins are in the cell. So thank you for your attention.
It's a pleasure to follow Jerry and to talk about the studies that we've been doing in C. elegans on trying to understand some aspects of how brains process information. We're guided by the complexity of the human brain and the understanding that that may be too much to understand all at once. And particularly what we're interested in are intermediate scales of time and space, the level at which circuits function and the level at which behaviors are modulated and generated over the course of minutes to generate things like motivation, emotion, and other forms of regulated behavior. We study these in the nematode worm C. elegans, a simple animal that's transparent. It has exactly 302 neurons and 7,000 synapses defined from its connectome over the course of many years of studies from a variety of different groups. And with this very simple nervous system, it can sense, learn, explore, aggregate, mate, and have other behaviors. And it does this using a nervous system that shares many genes with other animals, including humans. And the point I want to make today in the talk is often how things that we learn do seem to be circuit principles that apply across multiple animals, not just across one. And so there may be themes there, just as there are principles in molecular biology that can apply across species. So just very quickly, the 302 neurons are divided into sensory neurons, integrating neurons, and motor neurons. And also, there's a small cluster of enteric neurons associated with the gastrointestinal system. So in my lab, we have taken one particular set of behaviors as the basis for what we study, and those are chemotaxis behaviors. And we think that they're interesting. They've been known in the C. elegans field for a long time because they represent something more like a computation, not just an immediate input-output relationship, but some sort of a calculation about the environment. So this image on the right shows you a single worm chemotaxing across a 10-centimeter plate to an odor. That roughly to scale would be like you chemotaxing to an odor across this auditorium. And what you can see is that while it gets to the odor over the course of about half an hour, it doesn't do so all at once. It's a set of excursions, there are a set of different motor behaviors, it spends a certain amount of time going in the wrong direction, and yet at the end of the trajectory, it's reached the odor. And so this is a probabilistic behavior with a reproducible outcome. And it's doing this using a whole set of different behavioral maneuvers, of different actions that I'll mention as well. So we've been trying to understand how this works, and our framework for doing that starts with using genetics to identify genes important for chemotaxis, which is something Piali Sen-Gupta did when she was in my lab, using various kinds of genetic and molecular reagents to figure out how those genes were acting, where they were acting, and what they were doing for the behavior. And for that, we get the following sort of first-order understanding of chemotaxis, which is that there are many, many odors, and they are detected by many, many, many G-protein-coupled receptors. C. elegans has more G-protein-coupled chemosensory receptors than you do. It has more than dogs do. The only animal, to my knowledge, that has more odorant receptors than C. elegans is the elephant. And this large number of sensors is expressed in about 26 sensory neurons. So each one is packed with many different receptor proteins, but they're expressed in specific patterns that let them detect different odors. And so when Piali was in the lab, she found that one of these receptors, called odor 10, was required to detect one attractive odor, diacetyl, and it was expressed in the AWA neurons that mediated attraction. And then Emily Tremmel, a graduate student in the lab, showed that if she used genetic methods to remove odor 10 from its natural site of expression and instead express it on a neuron that normally detected repellents, the animals were now repelled from the odor instead of attracted. So we got a sense that we could define the chemical specificity using a receptor, and then we would be able to, but then the sensory neuron itself seemed to be wired in innately to particular behavioral outcomes that guide behavior. And this principle appears to apply also to, for example, the taste system in mammals, or certain aspects of the somatosensory system that distinguish different kinds of attractive or aversive touch. And by the way, I'll allude to work that was done by other groups. They will always be color-coded in black. And so, for example, the strategies for chemotaxis were worked out by the Lockery lab and by Yuichi Ino's lab in large part. So how does this work? There's some kind of a circuit downstream of the sensory neurons, right? And so we started with our 302 neurons. And in addition to the sensory neurons, Jesse Gray, when he was in the lab, identified a group of about 20 interneurons and integrating neurons that seem to represent the key circuit for foraging, spontaneous locomotion in a uniform environment, as well as chemotaxis, tracking up those gradients. And I should say that other people who did important work in this area include Ikoue Mori, studying thermotaxis behaviors. And so what Jesse did was to show that different neurons in this area are not required for particular motor actions, like reversals in general, but rather they regulate the pattern and the probability of reversals, or forward movement, or certain kinds of curving movements, or certain kinds of turns. So this is the place within the circuit that we have been focusing to understand how chemotaxis happens. So one question is, for example, we look at all these connections. We take one neuron here that detects attractive odors. It's connected synaptically to many of these target neurons. What does it do? What information does it convey? And here, Srikanth Chalasani, working with Nikos Kronos, when they were both postdocs in the lab, first used genetically encoded calcium indicators to track the activity of multiple interconnected neurons, then used genetics to define what the transmitter was of the sensory neuron, and then the transmitter receptors on its downstream targets. And in general, I'm going to just let this kind of technical work be in the background, but just to say that we found that the sensory neuron is inhibited by odor and activated by odor removal. And in that respect, it resembles photoreceptors in the vertebrate retina, which are inhibited by light and activated in the dark. And of its two sets of downstream targets, one set has the same pattern. It's activated by odor removal, and that set regulates reversal behaviors. And the other set has the opposite effect. It's activated by odor addition, and that set promotes forward behaviors. And looking at this, we realized that it actually had not just similarity at that first detection step, but for a couple of steps with information processing in the vertebrate retina, suggesting that there are shared motifs for circuit processing. That not only do the signaling mechanisms and even the signaling molecules resemble each other between the vertebrate retina and the worm nose, but these neurons use the same transmitters to either make excitatory or inhibitory connections to different downstream receptors, each of which is activated by glutamate. In the case of the vertebrate retina, the off bipolar cells and the on bipolar cells, to signal lights off and lights on, just as these neurons signal odor removal and odor on. And we found a couple of other circuit motifs that we think may be showing us some kind of conservation. A much more complex one that involves integration of multiple sensory inputs was characterized by May Dubashevitz and Chung Liu when they were in the lab. And that involves multiple sensory neurons that were characterized electrophysiologically as well as functionally, which have to be activated at the same time for a downstream neuron to become activated from a bistable state. And it turns out that very similar transmitters and principles are used in the fly visual system in motion-selective neurons that must also integrate very specific information from excitatory and inhibitory inputs to define preferred visual stimulus direction. And that's work from Axel Borst's lab. So here we have kind of a first-level understanding, passing one synapse. Now let's try to move from there to behavior. And this work was initiated by Dirk Albrecht when he was a postdoc in the lab, working in part with Johannes Larsch. He developed microfluidic devices that enabled us to put worms in a freely moving environment, expose them to highly controlled odor stimuli that we could vary the exact concentration and temporal pattern, and then monitor both their behavior and the activation of particular neurons, as shown here. So the animal is moving freely, an odor arrives, is removed, and the animal does a little reversal removal maneuver upon odor removal that's associated here with the activation of the AWC sensory neuron. So we can do this experiment now reproducibly many times, and at this point in the talk, we move from watching individual animals one at a time by graduate students and postdocs to using things like machine vision and computational approaches and to track the behavior and get a quantitative sense of its output. And what Johannes and Dirk found was the following. Although the olfactory neuron responded extremely reliably and was activated in every single trial, here for example by becoming active after odor removal, the behavior response varied a lot. There were 3 different behavioral responses, one very elaborate pirouette response, one shorter small reorientation, which is what you saw on the last slide, And then about 20% of the animals that didn't do anything at all. They paid no attention to the odors. So we've crossed from having a reliable sensory response to a very unreliable behavioral output. So how does that happen? Where does behavioral variability arise? Where in the circuit should we seek that? So a couple of people in the lab started to look at that, asking, was it the transmission of information within the circuit or the target neurons that were relevant? And for example, Donovan Ventimiglia, a graduate student, used fluorine synaptic vesicle release markers in a single neuron in vivo to look at the response to odor. And what he found is that reliably, every single time the odor was removed, these neurons released neurotransmitter. So the transmitter is being released by the sensory neurons, but the animal is not always responding. However, when Andrew Gordis, who's now at Hopkins, looked at the downstream neurons, he saw something very different. He looked at odor addition and removal, and he found that even one synapse across with that AMPA receptor I was telling you about, the neurons were quite unreliable. They only responded to odor addition or removal about half the time, or even a little less. So something was changing between these 2 points. And a really important clue to what that was realizing that these 3 neurons here, which were interconnected, were all unreliable in very similar ways. When one of them was activated, almost all were activated, and opposite when they were inactivated. There was some sort of a correlation to their behavior along with their variability in the output. And around the time this was being developed, work in a variety of other labs, not my own, recognized that in fact, C. elegans brains have collective activity states in which many neurons are recruited simultaneously or near simultaneously in a correlated fashion. And this idea has been emerging from other brains and was emerging around the same time. This example here is more extreme, but for illustrative purposes, it's a good example to use. It's from Saul Cato and Manuel Zimmer in Vienna. And what they found, looking at many, many neurons, including the neurons in that reversal circuit that I just showed you, is that many of these neurons are simultaneously active, that one set of them are active in reversal-like states, and a different set are active in alternating forward-like states, and then an overlapping set in turning. So what you see is that there are neural activity states and behavioral states that are correlated with each other, and that the system is moving through those in what you can call a manifold, whether neural or behavioral for behavior. And this turns out to be what the worm is interpreting to decide whether to let an odor stimulus through. What matters about whether the odor stimulus goes through is what the state of the circuit is when information arrives. Effectively, odor and the motor state, the circuit state, are competing to control the circuit and the behavior. And sometimes the circuit is accessible to the odor input, and other times it is really just on its own manifold, and it's not going to pay attention. And so here on a time scale of seconds, we start to see a switching behavior, an internal dynamics in the circuit that then regulates its response to different sensory stimuli. And I want to remind you that chemotaxis is probabilistic, that the behaviors were varying and were superimposed on endogenous foraging behaviors, which we think may be related to those ongoing activity patterns. And it actually sort of raises a set of other questions that I just want to sort of allude to very quickly, which it indicates that coordinating the activity of a circuit is a key circuit property, that getting the right neurons activated in synchrony with each other, or at least correlated with each other, matters. And in that context, Elise Sordillo, a graduate student in the lab, was able to look at the properties of different neurons in the circuit and find, for example, that this one here has a key role in coordinating a set of neurons that are simultaneously active. That it makes a set of gap junctions to other neurons that are active during reversals, and it makes inhibitory chemical synapses onto neurons that are active during forward movement. So here an on or an off state. But really interestingly, the gap junctions that it makes not only activate reversals when the animal is reversing and these neurons are active, but when the neuron is hyperpolarized, it inhibits the other neurons. So it stabilizes them in both their active states and their inactive states by coordinating their functions through gap junctions. And we think this is an interesting motif that we can look for more broadly. So I'm now going to make a switch From looking at this circuit and the way that it works on this second-to-second basis while the animals are moving forward, reversing, turning, and thinking about how it is acting on longer time scales and how it's responding to different kinds of contexts to generate different behaviors. And here I will be talking, as Jerry did, about neuromodulators, these small molecules that seem to create internal states that modify behaviors. In mammals, they regulate sleep, hunger, mood, arousal, social drive, pain, and stress. And they fall into two categories. They're the small molecule biogenic amines, like serotonin and dopamine, and the neuropeptides. There are just a handful of biogenic amines, but they are very well known as being the major targets of many psychiatric drugs, like SSRI antidepressants, and drugs of abuse, like cocaine. And similarly, the neuropeptides in the endorphin family are the molecular targets of addictive painkillers. There are about 100 other neuropeptides, not just endorphins. There are about 100 neuropeptides in worms and in humans, so it's a very complex system of comparable complexity. And interestingly, it's not so different between them despite the difference in the size of the nervous system. All of these molecules signal through G-protein coupled receptors. So they are not primarily regulators of neuronal excitability in a direct way of regulating ion channel. Instead, they have biochemical readouts, which will tend to be slower and modulatory at a cell biological level compared to the fast readouts of opening or closing a channel. And of course, these are well known in many animals. Probably the neuropeptide your family will have heard about the most is oxytocin, a neuropeptide heavily implicated in affiliative behavior across a variety of different animals. When she was a postdoctoral fellow in my lab, Jennifer Garrison identified an oxytocin homologue in C. elegans and found that it was also engaged in social behaviors, in particular the behavior of a male during certain parts of its mating interactions with a hermaphrodite mating partner. Similarly, there are other neuropeptides also linked to social behaviors, like the tachykinin peptide studied in David Anderson's lab. And like oxytocin, these are peptides that seem to have, in some ways, analogous functions across a wide range of animals, that the same family of peptides are involved in regulating aggression in fruit flies and in mice, territorial behaviors. So there's something to this idea of these molecules regulating behaviors. So not only mating, but other social behaviors in worms are regulated by neuropeptides, and they're context dependent. And we first started studying those in the context of a neuropeptide receptor that regulates aggregation, not aggression, but rather the forming of these sort of swarms or feeding groups of animals that gather together on an otherwise positive environment. This behavior is triggered by the presence of certain pheromones. C. elegans makes dozens of different pheromones, and it uses them to signal to each other in great detail about its sex and its age and its reproductive status and its feeding state and so forth. And those are what seem to be major regulators of this behavior. And we started studying this actually because there was a natural polymorphism where our laboratory strain tended to be very solitary compared to most wild strains, and that was largely due to the action of a neuropeptide receptor that had high activity in the laboratory strain. So this is, in a sense, an antisocial neuropeptide, mediates solitary behavior. And when Evan McCosco was a student in the lab, he identified the site of action of this as being involved not in the primary sensory neurons, but rather in a set of integrating neurons called RMG neurons that are outside of our main foraging circuit, but definitely of interest. So we wanted to understand how this behavior was generated. And this was a collaboration between Hee-Yeon Jang, then a student in my lab, and Piali Sengupta's lab at Brandeis. And what they did was to try to figure out, well, how do you get from this detection event to this aggregation event? And they identified sensory neurons that were important in detecting these aggregation pheromones. And they found that these sensory neurons were activated by the pheromones. And interestingly, they were activated both in the isolates or the genotypes that were avoiding pheromones or solitary, or in those that were aggregating. So something up to this step was actually quite similar between both of these two different behavioral outcomes. And they're mediated by different synapses, and that is the explanation. So the avoidance circuit is mediated by chemical synapses, the aggregation circuit by gap junctions. And what the neuropeptide receptor does is to basically come in and shut off the gap junction circuit and thereby shut off the aggregation behavior, enabling the information to flow down an alternative pathway, essentially rerouting information flow to generate avoidance through chemical synapses instead of aggregation through gap junctions. So what we're seeing here is that the role of a neuromodulator is to gate the access of sensory information to one of two alternative circuits. And this, of course, captures ideas that people have been uncovering in the role of modulators in many other brains. So this idea of regulating sensory access is seen in the mouse visual cortex, where mice that are running and highly attuned to visual stimuli have acetylcholine neuromodulators released in the visual cortex, and these yield a strong response in the resulting neurons, compared to stationary mice, which have a much weaker response. And similarly, in Drosophila, gating of sensory information accounts for why a starved fly is much more sensitive to sweet stimuli than a fly that is sated. Dopamine release onto the synapses in the starved fly allow information to be transmitted more efficiently to their downstream synaptic partners. So here again, a common motif across systems. And what this is showing us is, in fact, that each circuit is many circuits. That an anatomically fixed circuit, like a circuit that detects certain pheromones, can be part of two different circuits. In this case, it can be part of the aggregation gap junction circuit that I alluded to, that's regulated by neuropeptides. But when that is shut down, this sensory neuron joins in with the other sensory neurons into the foraging circuit and regulates those same avoidance behavior neurons to generate a behavioral readout. So now it's switched back and forth between these two circuit outcomes. A little more sophisticated than just joining or not joining, but same general theme. And this is a behavior that takes place on a time scale of a few minutes. And this longer time scale is what we really see when we start to look more carefully at neuropeptides. So for example, we can look at global arousal states of the animal, analogous to high energy or low energy states. Animals moving spontaneously around will spontaneously oscillate between low activity states where they stay in one area and reverse a lot, and high activity states where they actively move from one area to another. So this is an example of foraging behaviors that allow animals to explore environments in different ways. When he was a postdoc in the lab, Steve Flavell identified two neuromodulators that regulate these two different behaviors of dwelling and roaming. And they regulate them in an interesting way. They regulate in animals that lack serotonin, for example, dwelling states are much shorter. Instead of being about 8 minutes long, they're about 3 minutes long. But while the animal is dwelling, it's dwelling perfectly fine. This molecule is not required for the individual actions in a dwelling state. It's required to generate the longer-term, minutes-long behavioral state corresponding to a low-activity state. And PDF, a neuropeptide, does the opposite to roaming state. And these two antagonize each other to set the overall level of roaming and dwelling by changing state durations and probabilities. So here we're working on a time scale of minutes. And here again, Steve mapped out the source of the two neuromodulators and the source of their targets in a heroic set of experiments involving replacing the neuromodulators in all of the serotonin-expressing neurons, for example, individually, then looking at the different receptors and where they were expressed. And what he found is the following. The modulators are made outside our core wiring circuit. They're made outside the foraging circuit. Actually, serotonin, in this case, is predominantly made by one of those neurons in the enteric nervous system that's detecting feeding state. And so that's what's coming in to regulate roaming and dwelling. But the targets are within that core circuit, and that's what this yellow line indicates. This modulator acts on multiple targets within the circuit to regulate their activity. The antagonistic, roaming stimulating modulator acts on multiple neurons, only one of which overlaps between the two, so they're all being set together. And this is reorganizing the wiring diagram. And the modulators are not synaptically connected to any of their targets in this wiring diagram. This is a wireless diagram that is distributed and superimposed on the wiring diagram for foraging behavior. So slow and fast mechanisms are actually anatomically distinct as well as chemically distinct. And so here again, we have one circuit. It can be our standard chemotaxis circuit and taking information from these neurons here. Or it can be the foraging circuit and measuring information from modulators to change overall activity states. Or it can be the anti-aggregation circuit that is regulated by neuropeptide access of yet another sensory neuron to the same set of targets. Each circuit is many circuits on the time scale here of minutes in which it switches from one of these to another. For the last couple of minutes, I want to talk about one other aspect of behavior, which is learning. So for years, we've been thinking about how to teach worms and what we could teach them. And Yun Zhang, when she was a postdoc in the lab, was really inspired by work on conditioned taste aversion. This is the strongest form of learning in any animal. It's when you experience a neutral stimulus and then soon thereafter become nauseous or sick. After that, the neutral stimulus becomes aversive. And even years later, if you encounter that stimulus, you will reject it. You'll experience an immediate wave of nausea. Many of us have had that experience in our own lives. And for anyone who's interested in the history of science, I encourage you to read about how conditioned taste aversion was discovered. It was a real outsider science working at the Navy Research Labs in San Francisco, Hunters Point. And it's a great story, and it's a totally entertaining Wikipedia page. But in any case, if you can teach this to any animal, maybe you can teach it to worms. And Yun learned that you could teach this about worms if you used pathogenic bacteria as a signal that made them sick. So worms eat bacteria. Normally that's a good thing, but their environment has a lot of bacteria in them, and some of them are pathogens that will infect and eventually kill the animal. So this is a real feature of their environment. It's one that they have to deal with on a daily basis. And it turns out that they will learn to avoid the pathogen that made them sick. That with as little as 4 hours of training in the presence of a pathogen, in this case Pseudomonas aeruginosa, C. elegans, when then subjected to a choice between the odor of Pseudomonas and E. coli, switches from being actually slightly preferring the Pseudomonas at baseline, because it's actually there are a lot of Pseudomonas species that are good bacterial foods, to now radically preferring the E. coli to the Pseudomonas. So it's switching its behavioral preference in response to the odor. And this associative learning is specific to Pseudomonas and a couple of other species within that genus. It's not observed to other bacteria, and it can last for various periods of time. And this is something that the worms really want to learn. When Xin Jin was a graduate student in the lab, she found that if you exposed young larvae to Pseudomonas, they would avoid that pathogen for the rest of their lives. They would have an imprinted memory to avoid that particular bacterial odor. And other labs, notably Colleen Murphy, have actually shown that parents will transmit some of this avoidance to their offspring. So this is something that worms really want to learn. And that makes it possible to study it. And first Yun in my lab, and then Xin in my lab, and simultaneously Yun in her own lab, now at Harvard, have mapped out more of the circuit. And again, the learning circuit, this is a chemotaxis behavior, is modifying the chemotaxis circuit. It's coming in to some of the same systems. It's using some of the same input neurons, so the pathogenic bacteria are sensed by some of the same neurons that sense naive, attractive, or repulsive odors. But other neurons are now being recruited, like this serotonergic neuron releases serotonin to help generate the behavior. Serotonin, same modulator, different neuron from the one that regulated dwelling. And other neurons in the circuit carry out different parts of the task. Xin, because learning and retrieval of the memory were so widely separated in time, was able to show that the requirement in learning and in retrieval use different neurons. And so here we were talking about these modulators again acting, but now on a time scale of hours to days. Now, pathogen learning is one thing. It's one thing to just like the pathogen odor or dislike the odor. They will actually physically leave a bacterial lawn of pathogen and avoid it and stay off of it. This enables them to live longer in its presence and to potentially detoxify it. And this behavior is also a very long-lasting behavior and a very slow behavior. It's been characterized by a number of labs over the years. It can take up to about 20 hours for the animals to develop a full-on repulsion. And Javier Marquina Solis, a student in the lab, has been looking at how that occurs, and what he's found is that it drives global sustained regulation of behavior by and through the regulation of neuromodulators. So pathogen elicits serotonin as part of its signal. It also drives internal signaling that drives avoidance behavior through neuropeptides, and these regulate an antagonistic circuit with a different neuromodulator, tyramine, that Jerry mentioned. And here we have a case where there are antagonistic neurons, like there were for roaming and dwelling, antagonistic modulators, like there were for roaming and dwelling. But now, in addition to regulating release and detecting it through receptors, in this system, the transcription of the modulators is being regulated. The FLIP1 factor is turned up, the tyramine synthetic enzymes are turned down. And at the whole animal level, serotonin levels triple in association with this avoidance behavior, and tyramine levels drop by a factor of 10. So there's really a metabolic effect. This information is propagating into the nervous system from the body and then back out again. And that, again, just reminds me that to tell you that each circuit is many circuits, and also to tell you that there are other elements of this component of the circuit as well. Josh Mizell and Dennis Kim have done a beautiful job of characterizing a TGF-beta system that also plays a role. So when we look at these neuromodulators and we try and draw a Venn diagram of cells and modulators and behaviors, there's nothing linear here. There's lots of antagonism, there's lots of cooperation, but we can say a few things. It's orthogonal to fast circuits. Information is distributed among multiple neurons. The modulators are multifunctional. They're subject to slow regulation at the transcriptional level, and Mike Greenberg is out here, and I just want to remind you all that some of the first genes studied as immediate early genes whose transcription was regulated by activity were neuropeptide genes. And they seem to select among connections to reroute information flow, that they let the animal change its mind. So my conclusion is that circuits are flexible, that sensory cues provide information, that they are fed into an over-connected wiring diagram, and that neuromodulators, or the internal context, allow the animal to reversibly and shape its behavior in a context-dependent fashion. This work was done in my laboratory starting at UCSF. Here are pictures of the lab across the years with different lab members indicated. 20 years ago, we moved to the Rockefeller University. Here we are in our unrenovated lab, where we've been ever since. It's been a wonderful environment. They've been wonderful people to work with. And I just want to say that you never stop learning. And in addition to my graduate and postdoctoral mentors, I feel that I've had many mentors and collaborators throughout my career whose influence I want to acknowledge and who I want to thank in this room here. And I noticed yesterday that several of these are Gruber Prize winners. And these are the people who taught me genetics, who taught me chemistry, who helped me to learn how to record from these neurons using genetic indicators, theory, and also taught me some things about leadership and the importance of that in science. So thank you.
Thank you. Thank you once again to both of our speakers for outstanding talks on this fundamental neuroscience. We have time for probably one question each, and we have our moderators from our prize committee, Drs. Eve Martyr and John Monsell, who will facilitate the discussion forthcoming.
So I'd like to start with Jerry. We had several questions posted online about what other connectome efforts are underway, and I'd like to expand on that just a little bit and just mention that in both of your talks there was a lot of emphasis on how conserved the computations and the circuits and the behaviors are across species, and even some remarkably complex behaviors like mating or aggression. And I've met several people whose behavior could probably be explained by a male aggression neuron. But the real question is, in getting to understand human behavior, we're going to need to tackle some questions like understanding facial recognition, language, math, things that probably just don't occur in invertebrates. So The broader question is, what sort of efforts are underway in developing sets of connectomes from different model species or different developmental stages? And to what extent do you think we need to build a ladder of model systems and connectomes in order to get to understanding the human condition?
Okay, I think we need to build more connectomes of all sorts of animals. And I know this sounds daunting when you think about how much bigger the mouse is. But for here, I'd just like to point out that I started my career in genomics, and from the Genome Project, I can remember spending an entire weekend at an NIH-funded meeting, and our goal was to decide, should we do the connectome of a mouse or a human, because in the history of mankind, we'll only ever be able to afford to do one large genome. And now there are individual studies doing 100,000 human genomes. So I think what will happen is this will get cheap enough and easy enough. So if you were a graduate student there, I think you could look forward in your career to having 100 human connectomes from 100 different people. And that will happen because it just requires technology advances that we know how to do. So I think as it gets cheaper, we'll be able to have everything. That is my simple answer. And I didn't mean to imply that we'd understand all of human consciousness from studying a fly. Maybe only half.
Thank you.
So I have a question for Corey, which is partially motivated by a question from the audience and partially, like John, changed it. And that is, the question from the audience is, if you had a readout of the state of every neuron and the concentration of every neurotransmitter, do we have the computational capacity to predict the worm's behavior? And if not, what approaches need to be developed? And I would just like to amplify that with, you were talking about state-dependent actions, state circuits that change their state. And a lot of your genetic manipulations and perturbations may have variable actions as the state of the circuit changes, so you can get ambiguous answers under those conditions. So comments to those two.
So first question is, when will we have solved the worm brain? And when the connectome came out in 1986, it seemed like we would be able to use that to solve the whole worm brain right there with that. And I was at MIT at the time, and these people would come over. I think Marvin Minsky was still there at the time, various computer science type people. And they would come in and they'd say, we're going to solve the worm brain. It was really fun to talk to them. They were very smart. And then they went away and we never saw them again. And in retrospect, the wiring diagram was too ambiguous. There were too many possible ways to read it. We didn't have anything at the time then. And but now we have many more elements. And there's a lot of, we know the molecules, we know the sign, we know the transmitters, we know the signs of many of the connections. Not all of them, interestingly. And there's a big discussion right now about, as there has been probably every decade, as more and more information comes along, it's like now the time that we could really understand this. People are doing these whole brain recordings now. Resolution could probably get better, missing a lot of sort of interesting things that are happening on the margins, but maybe that's good. Modulators are getting there, but we still need to have ways of measuring the modulators and how far they diffuse and how stable they are, and how they interact with receptors of different affinities. The ongoing dynamics, I think, are going to be a really good organizing principle, as these sorts of manifold dynamics are in a lot of systems neuroscience contexts. And one of the fun things about it is that there have been some discussions. There's been a position paper written. I think Gal Haspel is the first author. And it started getting some really interesting people interested in the worm in ways that I find exciting, including some of the people doing systems and theory who weren't thinking about C. elegans in the past, like Ela Fiet and Scott Linderman. So I'm optimistic that we might be able to put together a group to do that in a more serious way, but we don't have it now. Oh, and in terms of genetic variability, it is true that when you knock out a gene, you're going to change the whole system. And, but I want to remind you of how much genetic variability there is between human beings right here in this audience right now. And in fact, one of the biggest impediments to human genetics for many, many years, until there were many, many human genomes sequenced, was that we just could not deal with the huge amount of variability that exists between normal, healthy human beings and their different genetic repertoires. So I think accepting, accepting some amount of variability is part of what natural selection does to generate lots of different individuals, of which the better ones are going to ultimately be more represented in future generations. Yes, some of the ones that we make are very extreme. You are not likely to find animals without some of these core molecules out there anywhere except a laboratory. But some of them, especially the modulators, they're just in a slightly different behavioral regime. And that's why I emphasized that they could do all the behaviors, they just spent less time doing them. And that might describe many people that I know.
And with that, I think it's a good time to end.
So I want to thank again Corey Bargmann and Gerald Rubin for wonderful presentations, and congratulations again on the 2024 Gruber Neuroscience Prize. And this concludes the Gruber program. So please join me in thanking them and congratulating them again.